Welcome to the 16th International Conference on Emerging Technologies for a Smarter World. Focused on machine learning, artificial intelligence, blockchain, and computational medicine, the discussions we have today will both shape and respond to our rapidly evolving world. To all of those attending, thank you for your participation in this critical work. And thank you to CWIT, the New York State Center of Excellence in Wireless and of 2020, and our reliance on new technologies will have long-lasting implications. I am proud of how Stony Brook adapted to remote learning this past spring and how it has, this fall, prepared staff, students, and faculty for a hybrid education experience that combines in-person and remote classes. When the pandemic hit our university and region, Stony Brook worked to implement new modes of telemedicine and research initiatives to help our community. We leveraged our unique history of collaboration and cutting-edge research to respond to the challenges that COVID-19 posed, from critical supply shortages to health and safety protocols. Just this summer, Kiru, a startup company out of the Center of Excellence in Wireless and Information Technology, worked to develop a machine learning tool to analyze and predict regions most at risk for COVID-19, which could be a huge asset for officials who need to act quickly and distribute resources. By fostering an integrated and multidisciplinary research community and collaborating with organizations across the world, Stony Brook University and the Center of Excellence in Wireless and Information Technology are in a unique position to respond to the massive technological disruptions and challenges of our era with critical thinking, invention, and social responsibility. This is an opportunity for us to look squarely at the future and build something bright and new. The International Conference on Emerging Technologies for a Smarter World is about just that, the future, with presentations, panels, and expert speakers from across the globe. To start off, I'm honored to introduce Professor Ada Yonath, who will speak on the genetic code translation machinery as a therapeutical target. Professor Yonath is the 2009 recipient of the Nobel Prize for Chemistry for her studies on the structure and function of the ribosome, and she was the first woman in 45 years to win this award. She established the first biological crystallography laboratory in Israel, and since 1989 has been the director of the Kimmelman Center for Biomolecular Structures at the Weizmann Institute of Science. Her other awards and honors include, though are not limited to, the Israel Prize, the Linus Pauling Gold Medal, the Albert Einstein World Award of Science, and the Erice Peace Prize. So without further ado, please welcome Professor Ada Yonath.
Am I supposed to start talking? Okay. So,
So yeah, now I'm back to normal. The computer blocked me. So it is my pleasure to introduce Professor Ada Yonath to all participants of the CERIT conference. Uh, Professor Yonath got a Nobel Prize for work on the ribosomes. She's a leading and pioneering scientist in the world, and it's our pleasure to have her here and to collaborate on some basic studies in biomedicine. Thank you, Ada, and we look forward to your participation.
So thank you for inviting me. Uh, I think it's a big honor to talk to people that maybe can do what I cannot. And actually, I slightly modified the title of my talk and also my talk itself, because since I sent the topic, the title, sorry, since I sent the title, we got some results that I think may interest the people here. So shall I start?
Go ahead. Yes, you can start. Is your
Okay. Do you see my screen?
very good?
Do you see my screen? Good. So actually, I want to talk about medical problems that are in need for what you guys know much more than me. AI, machine language, and so on. But of course, we start with protein biosynthesis that is a um, performed by ribosomes. So you all know that I don't have to tell you that the genes are in the DNA, that the letters of the genes are coding for amino acids. There are only 4 types of letters in DNA. They're coding for over 20 amino acids. But anyway, whatever it is, the information here within the DNA is unavailable. It is masked by the structure. So therefore, it's being transcribed to a similar molecule, RNA, messenger RNA in this case, that has also 4 letters. One of them is slightly different, but otherwise it's the same. But from the point of view of main chain, RNA can stay as a single helix, so therefore the information is available for whatever cell components that have to read it, transfer it, and so on. Then it can be translated by the ribosome to proteins. And this happens in every living cell, from bacteria to elephants, flowers, whatever. So the ribosomes perform 2 major functions, decoding and peptide bond formation. And they do it in all cells in the same way, sometimes faster or slower. But anyway, they can work very, very efficiently. They can make 40 bonds between the amino acids. They are called peptide bonds in 1 second, with a mistake rate between 1 to half a million to 1 to a million. So if you want to hear about me, I used to be a good student, the fastest in my class, because I was very poor and I had many, many jobs. So I had to finish the lab fast. I had to make a single bond, 1 bond. It took me 6 hours and I was the first. The ribosomes are making 40, up to 40 in 1 second. So they can make a protein of 200, 300 amino acids in a few seconds, or in more sophisticated organisms, a few minutes. And they work like a factory. The top floor is where the information is being read, and the lower floor is where the peptide bonds are being made. It means the new amino acids that are brought on tRNA are stuck or connected to the growing protein. This happens, this arrow should be inside. And then when the amino acid has been connected, the genetic code is progressing in one trimer, it means in one code, and the track, the protein, the tRNA comes out with the other tracks and ready for the next job. It all needs very little energy, which I showed here, and that's it. Actually, in life, ribosomes are made of two subunits. Small subunit is represented here by the top floor and the large subunit by the lower floor. And that's the way it works. The small subunit is where decoding happens. The large subunit is peptide bond formation. And the tRNA that on one side can do the decoding because it has an anti-codon triplet here to connect with the messenger RNA that waits for it. On the other side, it carries the amino acid that goes into the newly born protein. So it took a long time, but almost 20 years minus 2 weeks. And but once we got the structures, we sort of immediately understood the function. And I don't know why it doesn't start here. Here. Okay, so we made a little clip together with art students from Jerusalem. And what you can see here is messenger RNA reaching the small subunit. In the cell, the two subunits are separated. They come together only when they have to make a protein, when the cell needs a protein. And this all happens with initiation factors. In bacteria, that's what you see here. There are 3 initiation factors. Number 3 is right here, the blue thing. So the blue things make sure that the messenger is in place. And then the second initiation factor can bring the first amino acid carried by tRNA. So the factors, the blue things are coming out. The amino acid is here. The small subunit with the messenger is here, and the tRNA is bound. The large subunit can come and interact, make bridges, and now the active ribosome has been formed. And you can see it from the top side as if it's in solution. Amino acids are brought by tRNAs with elongation factors. The ribosome helps in, out, with these hands that are actually very heavy. Then the information is being read here, the decoding. And from the large subunit that we took away and left only the tunnel through which the protein comes out, you can see this part here. Now you see the whole ribosome again. The small subunit, the protein grew and starts folding alone or with chaperone. And it's here in the lower part of the large subunit, and the whole process continues until there is a stop codon, and then the release factor is coming instead of the tRNA and goes in and separates the two subunits. The protein can go to do what it needs, and all the other factors, all the other players are waiting for the next job. That's all. So in the clip, you saw the ribosome something not very well resolved, but actually we know the position of each and every atom. These are the bacterial ribosomes, what we did first. Now there are many structures of ribosomes from all types of organisms. All are made of RNA and proteins, ribosomal RNA. In here, the ribosomal RNA is in gray and each protein is in a different color. Small subunit does the decoding. Just to remind you, the large one does the peptide bonds. So there is a huge number of ribosomes that function in each living cell. Mammalian cells may contain millions, like in the liver, 4 to 6 million ribosomes in a single cell. Even in bacteria, the number of ribosomes when the bacteria grows can be between 80,000 to 100,000. Because they are playing such a vital role over 40% of them, of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. It's very clever. You want to get rid of a pathogenic bacteria, stop the production or disturb the production of proteins. So the natural antibiotics are actually natural. They are the weapons that microorganisms from one type are using for interfering with the cell life of different species. And we human beings take the proteins of which. I'm sorry. So anyway, now we know how all the antibiotics of ribosomes are working. I must say that I didn't expect it, but it's nice to see. They are so small, I didn't expect to see something so small on this huge ribosomes, but we do see all of them. So this is adenine, very clever, very small and very clever, made in Poland, discovered in Poland. That's it, just doesn't let the messenger go. No messenger, no protein biosynthesis. Second one is tetracycline, much more useful and even smaller, occupies the position of the second tRNA. That's it. So these two are working on the small subunit. Let's look at two that are disturbing the large subunit. The first is erythromycin that came into usage just after penicillin. The first on ribosomes, and you see it's at the entrance to the tunnel. So a little bit of a protein, 5, 7 amino acids are being bound, but that's it. And the last one that you will see, clindamycin, is really the cleverest chemically. Remember, the peptide bond is being made here. It just sits in the bond, doesn't let the bond being made. So this is the way antibiotics work. I'm sure you know most of it, but still I want to continue a bit. And all the antibiotics, ribosomal antibiotics that we know are disturbing functional operations. They sit in functional sites, like in the decoding, in the bonding, in the tunnel, in the hinging, these motions. And some are sitting also here. I didn't show it in the end at the entrance of the tRNA. So once erythromycin was found, companies made all types of chemical improvements of them, and the whole family is called macrolides. And they're sitting, if you remember from the movie, at the top of the tunnel. So if we look at the tunnel this way, we cut it here, and this is what we see, the walls of the tunnel, which are the pocket where the macrolides bind. It's made only of RNA, ribosomal RNA. These are all types of macrolides. You see some are blocking excellently, some are blocking very good, some are blocking good. It doesn't matter. All block to very high extent, and all interact with one of the nucleotides that are making the wall, which is in position 2058, according to E. coli numbering. It's an adenine here. All interacting with it. So the way of the interaction, if we look at erythromycin, sits here, and A2058 is here. I must say that the chemistry here that was done by bacteria, without bioinformatics, without crystallographers, without anything, excellent affinity region, just perfect. So that's the way all the macrolides bind to the tunnel at this position. The difference between tunnels of bacteria and tunnels of higher organisms is just instead of adenine, there is an addition of NH2, guanine. So that's it. When there is here, just this is the difference, NH2. The distance here is too small, and there is no binding. So that's the selectivity. That's the way we can use the macrolides, because they won't bind to any organism that has G, which is everything over above bacteria, above prokaryotes. All eukaryotes have G instead of A. So after we understood how antibiotics work, now we want to look at something which is one of the most severe problems of modern medicine, which is resistance to antibiotics. So I don't want to talk much about resistance, and there are many ways that resistance is acquired, mainly by, mostly by mutations, random mutations. Once there is a good mutation, it's kept by the bacteria. But I want to show you the basis of one of them, which is actually based on selectivity. So if you remember, in bacteria, it works like this: adenine and erythromycin. And in human, it won't bind, or in animals, or in anything about prokaryotes, in all eukaryotes, it won't bind because there is here guanine. This is the rule of selectivity. So it's not such a big deal for bacteria to acquire this type of mutation, and then it is resistant. It's not the only way, and I really don't want to talk about all ways, and I don't know all the ways. This is one of them. So in order to fight resistance, which is a problem that is not very successful, and probably will never be very successful, because bacteria really want to live. We try to do something that may get out the humanity from resistance. And we decided to determine the structure of the ribosomes from the pathogenic bacteria, those that have resistance, and compare them to harmless bacteria, and maybe from the difference to come up with a new idea, which we did. So we got lots of lessons from this comparison, and I won't show you all of them. I actually will show very, very little. But the main thing is all bacterial ribosomes are similar. And please pay attention, similar, not identical. And that's actually the basis for what I tell you, going to tell you next. So lesson number 4 was beyond my expectations. It's novel antibiotic binding sites. And let me use a few minutes for this. So if we look at the superposition of a harmless bacterium in gray and a pathogen in cyan, you can see that the difference is not large, and there are also minimal positions. But when what I'm showing here are only regions that the pathogen has an extra piece, and it's on the surface, on the periphery. So you see there are some of them. And in the beginning, when we started this, people told us, hey, it won't work, only 8% difference. But the 8% difference is well ordered and well distributed, actually well positioned. So if we look at them in higher detail, I'll show you one. So you can see this is the addition of the bacteria, pathogenic bacteria. The rest is the same, exactly the same. But if this is exposed on the periphery, in a place that is far from the active site, and it has a structure which actually can be targeted chemically, why shouldn't it be a potential binding site? So essentially, we found many potential binding sites. I talked about this one a minute ago, but all these are also potential binding sites. And in a model system, we identified 25 potential binding sites, all of them new. We blocked them individually, one by one, and 16 of them, when they were blocked, they inhibit protein biosynthesis, not by binding to the active sites, but by binding to other components in the cell. And since these are not involved in primary ribosomal activity, so far there are no genes that are containing some type of machinery against them to modify them. So we think that this can be targeting for new next generation antibiotics, and we can design them the way that we want, or at least within the limitation, but it's in our hands, and we can then make them degradable and environmental friendly. So I didn't talk about this, and I want a minute for this: selectivity, similarity, and environment. So selectivity, it's really important that they are selective against the microbiome, which the currently used amino acids antibiotics are not. And I'm sure you know about the microbiome that resides in guts and hairs of the skin and lungs and so on. Because we can make the antibiotics the way we want, we can make them such that they won't harm the microbiome. And about environmental considerations, so most of the ribosomal antibiotics, I think now we can say all, have internal pieces, internal cores, which are not digestible, more important, not biodegradable. So when a person or an animal are using antibiotics, this dies, even if there is no overuse. These are coming out of us, and because they are so small, the purification systems may miss them, and then they can penetrate into the irrigation system and come back to us and increase the resistance. So when we plan our antibiotics, our next generation antibiotics, we can make them from nucleic acid or oligopeptides or combination, things that are not poisoning, things that are biodegradable, and it's all in our hands. So it looks like everything is there, but no company wants to take it over. So let's talk about something else. This was just an introduction to the problem that I want to suggest to you. So once you know what is the ribosome and as it works, in human or in mammalian, there are ribosomal mutations. Ribosomal mutations were found to be associated with diseases like cancer, red anemia, and others. The type of association is not known yet. I'll come back to it. So for instance, here you can see a nice triangle that tells you about which ribosomal proteins, which mutations in these ribosomal proteins can be associated with this type of cancers. Breast cancer, the biggest, but colorectal are all here. And the ribosomal proteins that are mutated, actually, I'm not sure that all of them are mutated, but their genes are mutated. All what's written here is genomic information. They are associated with these diseases. And as I said, also with anemia. So for one type of anemia, which is called diamond black thumb anemia, DBA, all these genes, look, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 3, 4, 5, 6. 26 ribosomal proteins were found, the genes of 26 ribosomal proteins were found to have mutations that are associated with this anemia. And actually what is known today, that each patient with this disease has only one of these mutations. But this is the second problem that is of interest. How are they involved? We don't know. And I want to show you some data about them. So first of all, these proteins are distributed on the whole ribosome. You can see here two views of it. Small subunit, large subunit, doesn't matter. Each of these is another protein. You can see them on the surface, but all of them have extensions that drill into the structure itself. And you can also see, if you don't want to see this type of presentation, you can see them as the lines, and then you can see how they really penetrate into the structure. So they're all over the place, all these ones. If we look at it in more detail, here is one of them. It's called 19. And you can see that it has so many mutations. Most of the protein is mutated. And when you look at it, these are the interactions that it makes with the ribosome. And pay attention, it goes into. You can see it here. That's all what we know about it. We know all about these mutations and where it sits. We don't even know that these mutations are expressed. We think so. But we know that these mutations are in the genes. You can see it here, and you can see here even the type of mutations. So there are frame shift, there are nonsense, there are deletions or insertions, missense, and stop gain at the end. And what we also want to show here is that these mutations, if they are expressed, they can interact with the RNA in all these positions, and this can propagate to other sides of the ribosome or to the surface or whatever. It means it doesn't say that if they hit a bad position, it's only this position that will suffer. Because if they move this part of the RNA, this will move another part and so on. You can see it here again. This is a protein that is mutated in cancer. This is the protein. You can see here its structure. And you see in how many places it interacts with the RNA and can move it. So despite the impressive development in understanding DBA, DBA was identified in 1936, almost 100 years ago, 84 years ago. And I think 2 years later, it was even not only identified, but categorized as connected to mutations in ribosomes, although they didn't even know what are ribosomes. There are still many open questions for you, for people that maybe can look at it not only as a structural biology, but in mathematical tools. So patients that have DBA and also several cancers, they can undergo self-renewing tissues. It means if the tissue is disturbed in anemia, it's either the bone marrow or the blood cells, it can recover itself, self-renew itself. How can this be if most of the ribosomes, and DBA is a dominant disease, either combinations of malfunction or just small number of ribosomes that escape the dominance? How can this be? There is no clear explanation for tissue specificity. Why does it go only to the bone marrow? We tried to find out if there is a common denominator among the various mutations. couldn't find anything logical, but maybe we didn't have the right tools for this. Also, there is no clear linkage between the clinical syndrome types and the type of mutation or their location. We couldn't find, we found only the correlation that there is mutation, there is a patient, but not more than it. So finally, we are still searching for an answer for the most fascinating query. How can such small alteration, such small mutation, each patient has one mutation, how can such small alteration cause such significant mental problem? And how can it do it only on one part of the body? Because all cells need ribosome. How does it express only there? So this is still open, and it really disturbs me that I don't understand how it happens. Let's talk about another molecule, collagen. I'll show you the connection to ribosomes, but this was my subject for my PhD thesis. At that time, when somebody said collagen, everybody said, "Call what?" It's not interesting, but now collagen is celebrating in cosmetics, in surgery, in many others. So let's look at collagen for a second. I want to show you something really nice that was done in the Weizmann. Collagen can shrink to two-thirds of its original size if you put it in lithium bromide. And bromine is the biggest attraction of the Dead Sea in Israel. So the Kachalskis, two very important professors, they are not with us anymore. They thought that they want to use bromine. And because collagen can shrink to third, they made here a machine. There are two baths, one water, one sodium lithium chloride. And these are collagen string between them. I hope that you can see. And this machine is moving on its own. It has its own rotation because it works on the difference in the baths. One has lithium bromide in it and one water. But by the time the water is already full with lithium bromide that was taken by the ropes, the machine stops. But this is just a side story. This is not the real story. The real story is the connection to ribosomes. So we prepared a controlled machinery to deal with surgery. So what we did, this is the collagen. We made of course, the recombinant collagen, and we made it circle. And every so many angstroms, we put in it cysteines. You see here and here. To the cysteines, we bound a little piece of peptide, polypeptide. And to this, one can bind the messenger of the protein that is needed. When is it needed? After surgery, when one wants to recover the collagen or the fibers there, that they will grow faster. So to put here the messenger RNA of the factors that are needed, growth factors that are needed, or for wounds and so on. So we did it with a Chinese postdoc that she even got a patent for it, and a cover page of Chem Biochem. You see here the collagen, and here comes the messenger, and the newly born protein, the good one that we need, is coming out of the tunnel. So how is collagen built? It's built of three strings that have the overall sequence of G, glycine, X, Y, N times, and N is very, very long. Usually, or mainly, X is proline, and there are also several hydroxyproline. But this is really not needed everywhere. The G the glycine is needed. So it's triple helix with helical parameters of 3/10. And you can see here each of the chains is colored by a different color, so it will be easier to see. From top, if you want to look at the contacts, all the internal contacts are made by glycines. These are the free chains, and you see here the glycines. So the glycines are stacked one on top of the other and make the contacts. This is the structure of all collagens in the body, in bodies of many mammalian. I think almost all mammalians, almost all eukaryotes. So that's the structure. How is the structure kept in nature? In nature, it's kept, I will show you in a minute, but in this machine that I showed you a minute ago, we put at the ends globular parts that like to fold on each other. So we called it fold-ons. Fold-ons on this side and on that side. But in real life, these fold-ons are called procollagen. They are on the N-terminal and on the C-terminal side, and they are globular, among globular pieces that keep the collagen in place. And nature takes care of it. And there are all types of collagens. And we talk now on the disease that I want to discuss is collagen type 3. There is 1, 2, and 3. Not so important, this is type 3. So just to show you, there are many structures of collagen, so we could understand the structure really good. And there are all types of different collagens are either made in the lab or in companies or in different animals. And you can see here if they are synthetic, large, high temperature, good or bad, dissolving, it doesn't really matter now, just that there are many and we can look at it. Let's focus on collagen 3A1, alpha 1, essentially. Here there is a paper from 2016 that cites so many papers talking about mutations in this just fibers that they are associated with melanoma, which is cancer. Here, there is another paper, which is really very impressive, that talks about the survival of this disease, Ehlers-Danlos syndrome, which is EDS, type 4, there are even many types. Mid-age of life is less than 50, and it's just collagen. They actually talk about lots of mutations. In one place, they talk about 600, in another type, over 500. And actually, the mutation we talk about for EDS is instead of glycine in position 1032, it's a valine. That's all. That's only one mutation. So I just want to show you their list of mutations. This is page 1. They have 8 pages like this. Just mutation. And at the 8th page is the one that I'm talking about now. So when you look at mutation, you can look at it from the point of view of the triple helix, or the connections between them, or the way they fold around each other. It doesn't matter how you look at it, there are mutations. So how do I try to explain what is the question? Let's say that this is the collagen, and here there is a mutation. So the triple helical structure is broken. How long it will take to have it back in place? We know that at the end it repairs itself, or not repairs, but builds itself again, because this collagen is found afterwards in tissues. It goes out of the cell. The 2 globular parts are being cut like everywhere, and it makes tissues, and the tissues are those that are deformed. So we know that this goes out, how long it will take it to recover itself. And it's almost at the end, this mutation. So in one paper, we found that it needs 20 rounds, huge part. But in our lab, we found something else. So what we talk about, let's say that this is the structure, it's exactly this PDB file. And here, this is the glycine that was replaced by valine, this one. So it bulges out. If we did it by the computer, we just model it. So it's this position. And also we don't know whether the mutation is in 1 or 2 or 3 chains. So even with 3 chains, this is the whole difference. And we found that within 2 to 3 rounds, it builds itself. Very, very different from what was found in the beginning. And I don't really know what's correct because neither of us could do it experimentally. It's just either computation or model building. So I just want to show you how it looks. This is collagen from different animals, different tissues. This is collagen. And we are going to look, if we want to look at the structure, we are going to look at these places if there are 2 of the rounds that are in different structure, or you can see here and here and here. So we have hopes, but I'm not sure that we will get it. And maybe computation can be more impressive and more to the point than we. So before I finish, I want to thank the Weizmann Institute that let me work. The decision was made by Michael Seller, although I was really slow and I was called Fantasioner and Bremer and lots of names. He was biologist, so they say that he understood it's important because of biology, but primarily that replaced him was a physicist, and he didn't kick me out. They got inspired by the advisory committee, and I was able to work in very strong collaboration with Max Planck, especially Max Planck in Berlin, and then afterwards in Hamburg, together with Dr. Weizmann, that his dream was to see how ribosomes work or how they are built, so we could collaborate really nice. He died 10 years before the structures came, so he didn't really get his dream. So I had 2 groups, one in Hamburg near the synchrotron, and one in Israel, and I want to thank all the members. This is the group in Hamburg that had angels, because they said only angels can work on ribosomes. And they came to the Dead Sea to look for bacteria. We actually used the bacteria from the Dead Sea for the first Christmas. This is the group in Israel. Most of the people here already left, only those that have stars. But I don't want to talk about everybody, just to mention that the real group leader is Dr. Anat Bashan. She's doing everything. And I want to show you Tamar. She came 18 years ago for 10 weeks. She's still around. She made a cake at the day that I took the picture. She had a birthday. You see the cake? Maybe you don't see well enough. So this is the cake. And this shows that ribosomes for us are considered sweet. I want also to thank my family. I cannot show you all of them, just my daughter who is an MD, and her daughter that is now studying medicine. But when she was really young, I got a prize in Paris, and she made a speech about me. And she said, as you know, she is a very busy scientist. That's about me. But she always finds time for me, and she gave me a prize. No, this is the Nobel Prize. What she gave me is this. The grandma of the year is Ada Yonath. And when I asked her which year, every prize has a year, she said, which year? You have to reprove yourself every year, otherwise I take it off the wall. So it's still on the wall for the last 13 years. So ribosomes became popular in Israel. This is a carnival. He is me, and she is the ribosome. And also young children became ribosomes. And Michel Kishka, one of our most admired artists and writer, he made me head full of ribosomes. And if you look carefully, small subunit, large subunit, small subunit, completely symmetric. Thank you.
Ada, thank you very much. It's our pleasure to listen to you not only as a scientist, but as a human being, as a good mother, and as a good member of our society. So we're looking forward to continue our collaboration. We began highly powerful computers. The size in the body is going down and down all the time, but the computation is going up and up. And we look forward to do wonderful things, so to see you accomplishing many, many important items in the body. In the next lecture, probably you'll tell us about the only living structure that you ever met at the Dead Sea. In the meantime, thank you very much, and we'll be in touch. Bye-bye now.
Bye-bye. And thanks again for the invitation.